cope not to bore you with questions that you are not interested in. But, sorry?
And they didn't Grant make some analogy between the immune system and ecology?
There is an analogy. There is an analogy, but no more than that, as far as I know. Yeah. Anyhow, this is also the reason why I use this kind of broad, or let's say unfocused title, because I wanted to speak about some models that has been suggested in the last decade or so to model species-rich ecosystems at relatively large spatial scales. Okay, and these are called basically macro ecological patterns. And usually, when we say species-rich ecosystem, we mean basically hundreds of species, hundreds, if not thousands of species. So you have to have in mind kind of a tropical forest, coral reefs, the Amazon forest, this kind of richness. And this also helps me explaining some open questions in the field that, okay, if you could help, that would be great. Before starting though, I have this caveat here. So it's important that I tell you from scratch that here I'm not going to talk about macroecological patterns in for multi-trophic ecosystems. So not species-rich ecosystem in which species interact in multi-trophic levels, like predator-preys, or mutualistic, or competition, or kind of these things. Simple one trophic level. The reason for this is not because I'm particularly fond of this, but simply because we don't have yet the machinery that is able to quantify in a correct way the what's going on at large spatial scale when we have really a lot of species that are interacting with each other. Yeah.
So Sandro, can I ask something?
Yes.
Can you give an example of,
yes,
of a one trophic community?
Yes.
Yeah, okay.
Now I'm giving all this.
Okay.
So, but I wanted to make it clear from the beginning that we are talking about these models, and this ecosystem with one trophic level are basically those that comprise species that compete all for the same pool of resources. So we have species of bees, species of butterflies, species of hoverflies, species of trees, right? These are one trophic level ecosystems. So basically, these are the empirical model systems for complex systems for which we can say something. We can introduce some simplified assumptions, we can do some analytical work that is useful, so we can make some prediction, we can make some comparisons and fit with the data.
And would grant applicants be a single trophic level?
Sorry?
Would grant applicants consider a single trophic level?
Probably, I think so. I mean, for the same code. Yeah. So these are a kind of examples, and specifically in this talk, I will say more about the tropical forest. So vascular plants that we can observe, we can measure on tropical forests or coral reefs, but where we can find hundreds of species like this. An example is this. This is one of the most famous examples, the Barro Colorado Islands. This is a 50-hectare plot in Panama, where basically each and every individual has been labeled, so we know the species of this individual, the spatial location, we know the diameter at breast height of this individual, and we know all this information at different years since 1985. Okay, every 5 years. This is one typical example, but we have even other examples like Paso Forest. We have Korup Forest. We have many examples of this, and this represents a huge amount of information that against which we can say we can see whether or not our models work, and this is what I will be talking in this. So just to say, so this represent so the trees in this tropical forest in Parma are a good example of one trophic level ecosystem for which we study these macroecological patterns. How do they look, these patterns? So we have patterns like this. So we have species-area relationship. So basically the mean number of species versus the area. So we tile all the plot with a given subarea, we count the number of the species that fall in, and then we take the average, and we have this increasing like behavior of the pattern. But here we are not interested in individuals, just species. If we want individuals, we have the species abundance distribution here. And here we have the number of species against the log of number of individuals, and we have this hump, more or less in the middle, in this log scale here. And this is also the reason why this pattern has been usually modeled with a log normal distribution, even though there is no compelling reason why we should use a log normal distribution. But yeah.
So what I was, I mean, I can sort of understand what's going on there.
Yeah.
You sort of take a radius, you look how many species,
Yes.
bigger.
Here it's an entire plot. So you put everything together. So you look at your plot and you say you see, you count how many species have any individuals.
It's confusing the first time you see these plots because you expect it to be plotted the other way around, a kind of rank abundance distribution. So what you've got to get is on the x-axis you've got bins containing numbers of individuals, and on the y-axis you've got numbers of species. It takes you a while to get your head around what that actually means. It does make sense.
Yeah, no, I mean, you can see that this is the probability that a species has any individuals in a log scale.
If you're thinking about picking species at random,
Yes.
but again, that's a bit weird because you think about picking individuals at random and checking their species. So
But this is a usual, I mean, people usually measure this.
no.
Yeah, and this is what, usually this is called a species abundance distribution. But, I mean, you can have it the other way around, like you say.
So this is how many species
Have
in this plot will have 10,000 individuals,
Yes.
or between 10,000
Between 10, yes.
and
So you see that there are many species with this many individuals here. So these are abundant species.
these are species that they all compete for the same
Yes,
thing.
trees, all trees.
Okay.
So here in total you have more or less 300 species,
Okay.
and you have hundreds of thousands of individuals. So here you have abundant species, and here you have rare species. Okay, and you have this peak in the middle, in the log scale.
Okay. Thanks.
Okay. So species versus area, species versus log of number of individuals. Another pattern is called beta diversity, and this is basically the probability that two individuals at distance d belong to the same species. So if I pick an individual here, an individual there, what is the probability that they belong to the same species? Okay, and this is one example of this plot. So the idea is try to model, try to make sense of this pattern.
And can I ask something again? So, sorry for asking a lot of things, but I'm trying to understand what's going on.
That's normal here.
So these are error bars, yeah?
Yes.
So why the error bars are so big as you go down? Because the relative scale, I mean, it's not, I mean, I just go from 0 to 0.
No, you're right, because here, okay, in here, they have lots of, they are looking within the plot. So here we are looking at the 50-hectare plot, only just within here, and here we have lots of points, so lots of good statistics. In here, those with this large error bar are just points that are outside and so they didn't survey everything. They have just some points outside the 50-hectare plot, and they try to do these measurements at large scale, but they couldn't survey everything.
Okay.
And so that's the reason why they have this large error.
But these are actual data. I mean, you know,
Yes,
one
yes,
has
yes,
one
yes,
has
yes,
counted stuff.
yes. All these are actual data for this BCI, for this plot. Okay? So, okay, if we look at other kind of one trophic level ecosystems like butterfly, birds, and whatever, the species area curve is kind of power law. So the mean number of species versus the area is, at least in the literature, is approximated by this power law distribution, even though there are no models that explain why. And even the fit, as you can see, is not very good. It's just a line that goes through the point. But I wouldn't trust these kind of fits. Another interesting thing that one could ask is that, okay, what happens if we put at random individuals in space and then try to, for example, calculate the species area relationship? Okay, so we have this 50-hectare plot. Let's put at random all the individuals, uniformly at random in these individuals, and then let's calculate the species area relationship that from this model. What you derive is this, where Ni is the abundance of species I, and S is the total species that you have in your plot. Okay, if you do that and you plot this in this chunk by forest, you get this red line here. So you systematically overestimate all species at all scales. Okay? Whereas your power law distribution here, I don't know if you can see that there is a blue line that goes on top of the black dots here. That is given by it's just a best fit. So let's say to a certain extent this does a good job in fitting this, and we cannot say that species are that individuals are distributed at random. There is something more, and today we know what is this.
So in particular, the Snowperson model does not have a power law relationship between S and A, right?
Yes. We know that basically this is due to the fact that species are aggregated in space. They are not randomly distributed, and we know that. But this was unknown at least 20, 30 years ago.
Doesn't that relate also with the previous graph that you showed us?
Yes.
With that so if you were to have that probability, you had, if you go to the,
Here.
couldn't you calculate that using the beta diversity?
That's a very good point, and actually that is one of the open questions today. So how can we go for, and I will show much of the work that I did and other people, is how to go from this from the other one. But you cannot do because this is just a 2-point correlation function to a certain extent. And here you don't have all the information that you have, for example, in here, because this involves a non-local measure. The other is just a point distribution. So you need more information, more than that. But we'll see later on this.
What's little a in your formula?
Capital A is the area of the entire.
So that S equals C A to the Z should be S equals C little a to the Z? If I compare that.
Yes.
Okay.
Yes, sorry, yes, sure. So here I have little a, here I have capital A. Okay, so another thing, so here I have this power law here, and if we go back, we have this. Okay, it's not strictly a power law, but you can. It's difficult to say with this linear relation. But here you have a power law. If you zoom in this log relation between the species and area, you see this triphasic SAR, species area relation. So from small scale to large scale, you have this sigmoid curve, and this has been fairly recently confirmed in this paper for different amphibians, birds, and mammals. So here, ecologists refer to alpha diversity, gamma diversity, and here beta diversity tells you the slope is related to the slope of this triphasic somewhere. So if you have the turnover is high, then the slope is high, and the other way around. So all this is a kind of phenomenology of this pattern, what's going on at large spatial scales in these species-rich ecosystems. Okay, now let's put some more flesh on these bones. And if you look at this one trophic level here, ecosystem, even though at different degrees, but what has been observed is that if you want to, if you look at them from, if you have a coarse look at them, so from a large spatial scale, then individuals belonging to different species are basically independent. That doesn't mean that there are no interactions. There are, and they are important. Even three-body interactions are, some of them are important. But if you want to model, if you want to look at these coarse scale patterns, then you can start with this first rough approximation, okay, for trophic. So because of this, people understood how, for example, how to model the species abundance distribution that I showed you before. So basically, they said, okay, we can use a simple birth and death model here, in which birth and death do not depend on species levels, and each realization of this process is basically a new species. So with this birth and death rates, basically, which encapsulates even a mild effect of density dependence when species are rare, you get this steady state here, and this can do a pretty good job of explaining the data that I showed you before. So in here, if you apply this, if you use this model, you compare with the data, you get these results here. So indeed, the model, this steady state is the red line. So the best fit of the model that I show you to this data, to these different tropical forests for the species abundance distribution, gives good fits. Not only that, and here this is BCI that I showed before, but these are other tropical forests in completely different geographical regions. So they don't communicate to each other, they are completely independent. And the interesting thing is that this X here, X and C here, X is always close to 1, very close to 1, and C does not vary much from forest to forest. So people at this point thought that, okay, here, because we have such a similarity of behavior, there is probably some general, some universal biological process from which these patterns emerge, okay? However...
Sorry, Sandro, I want to make sure I'm understanding. Can you go back one? This birth and death process is modeling the number...
So this N is the number of individuals of any species. So the birth rate of any species.
Sorry. It's the number, say that again, what is N?
Is the birth rate of any species in your system. And D is the death rate.
So all species share the same rate?
Yes.
They are all identically distributed?
Yes. Sorry. So, that's the reason why I'm just applying what I've told you before, the fact that they are roughly independent. So in general, one expects that there is B of I here. Yes, there is an index that depends on species, but because we know that roughly speaking, to a certain, to a first degree, we have independence, then we say, okay, let's get rid of this.
So for any given species, you're just counting the number of particles or trees.
Yes. So you see, when n is small, you are basically looking at rare species. When n is large, you are basically modeling abundance.
So when n is 0, you still have a positive birth rate.
Okay. When okay here. No.
Integration of trees.
Here, okay, where there are, if this represents, if you apply this to the entire system, then n starts from 1. For example, here, you see, when c is equal to 0, this does, you cannot start from 0, right? So if you look at the entire plot, basically you can only see, because you sampled everything, you can only have species with at least one individual. So this is a normalization constant that takes this.
So, sorry, I'm a bit slower than everyone else. So, n being 1 and n being 2 means what? What
Species with 2 individuals, species with 3 individuals, species with n individuals.
You're
So
essentially getting a probability.
this is the number of species with n individuals.
Okay.
And it's proportional to the probability distribution of
Yes.
just a single population
Yes.
evolving under
Yes.
those rules.
Yes.
So you have to modify somehow your grades, right? Otherwise you're absorbing.
Yes. But in here you don't have any absorb because C.
Right away.
See, you have the problem that you are mentioning is
Oh,
when C is 0, then you have the problem. But in that case, you modify. So you say that these are the rates. These are the correct rates when n is 1 or above, and when it's 0, you modify the rates.
Just kick it back to 1? Or?
Yeah. No, you say that D0 is 0, B0 is B tilde, and that represents immigration. And if you do that, you don't go into an absorbing state and you get the Fisher-Log series. And by the way, the Fisher-Log series was one of the very first models that were used to model this, but only from the statistical point of view, regardless of any modeling idea, regardless of any model. So it was just a distribution that fitted well the data they used.
If C equals 0, and if you left n equals 0 as absorbing, would that log series still be a quasi-stationary
Yes.
distribution?
Yes.
Should it be a
No, sorry. If C is 0, the quasi-steady state is an exponential. It's different. Sorry?
I've cleared up my own question.
Okay, good. I have to go quick, otherwise I can't say. So-
Sandro, if you don't necessarily have to finish today, you can give.
No, fine.
We've done that with Grant as well. He started with the talk. He was supposed to talk for an
No,
hour.
because the interesting stuff comes towards the end.
I'm just saying there is no reason for you to
No,
ask
okay, but I mean, this
something
is all very well known, okay?
to you, not to the rest. in some
Okay, in the
sense.
field, I mean,
Yeah.
whatever, okay? So this is the approach, so, am I clear so far? I mean,
Yeah.
Okay. So this is a model for the species abundance distribution in these species ratio systems, okay? So what I was saying is that, okay, from the idea was at the beginning, the idea was this, okay, now everyone is happy because we can model with this simple model here, we can explain tropical forest. So from this we can infer the biological or the ecological process that is driving this. No. Why? Because people found other models that can do more or less the same good job. So and this is the reason why I plot here the blue dots. This is a completely different model. It's called Hubble's neutral model that you start from different perspectives, different ideas, but in any case can explain the data equally well, I would say. Okay? So, and today we have at least, I would say probably 10 models that can explain this data with different ideas, so basically if there is an underlying biological process that is driving this, then it's something that we don't know yet. We probably, I would say probably it is not even a biological process. It might be something geometrical, something that depends on sampling issues. We don't know yet. So, but there are later on I will say more about about this and about how to improve these ideas. But anyhow, what is still remain is that the data here, the parameters are basically all are very much similar, even though forests are really scattered around the globe. They don't communicate to each other. So here, of course, we have different number of species, the absolute number of species is different, but if we rescale, the qualitative behavior is pretty similar. Okay, here we have everything is being filled, we don't have space. Now let me tell you more about this, this spatial turnover of species. So in this case, we try to we try to say more to understand what is the probability that two individuals belong to the same species when they are located at a given distance. So one of the, even here we have lots of models, but one of the most successful has been this voter model with speciation. So the voter model has a long history that dates back to the 70s with work by Liggett and others. And we knew that, for example, the voter model in 1D and 2D has an absorbing state, so we have monodominance, but in 3 and above we don't have monodominance, so we don't have this coarsening anymore. So people try to modify this old model, introducing this speciation. What is this? So basically the model works in this way. It's a really simple model because we want to get the idea of to get whether or not there is something that is general going on here. So we have a regular graph, and at each every side we have one individual, and we don't have any empty space. So population is proportional to area. And here the dynamics follows in this way. So we pick at random one tree, then with probability 1 minus nu, we substitute this with one of the nearest neighbors with the same probability, okay? And with probability nu, we substitute this individual with a new individual belonging to a species that does not belong to any species that was already present in the lattice, okay? So this is, one could say also immigration, okay, but usually people think of this parameter as speciation, okay? So in this case, by introducing this, this parameter here, you are preventing your lattice to go to a final absorbed state. And you always will have a non-trivial steady state. If you run this, dynamics here with the nu very small, and this is basically this is what you want, because basically a small you want something that is very small, because you're otherwise you every time step you have a new species, but this is not the case, right, in the real world. So if you run this and you go to stationarity, you get this kind of pretty figure. So this, and we know a lot about this voter model with speciation, in the sense that we know, for example, that the characteristic size here is proportional to 1/square root of the speciation. So we know, and we can rescale this in such a way that we find the same features. So basically, you can, if you use finite size scaling, you can relate species area relationship and species abundance distributions. You can relate at different scales. You can have adimensional variables that gives you hints about what are the basic features of your
Different colors are different species?
Yes.
I have to say, I find it amusing that when I see the traditional presentation of the voter model as opinion dynamics, there's typically not a term for the introduction of novel opinions, right?
Yes.
But do you have, I mean, do you sort of, I mean, will this system, does it have a true equilibrium state in the sense that you can say on average there are going to be maybe like 20 different opinions or something? Can you calculate the average number of opinions as stationarity?
not for this system. I mean, you can. I mean, it fluctuates, and yes, I think that you can calculate, but now I don't know. I don't know the answer. I think that you can.
Maybe I misunderstood. New, when you flip your coin and new comes up,
Yes.
positive.
Yes.
You take it from one of a set group of species or a brand new species? I thought it was
Brand
from-
new species.
Oh, not from like just one of the other
No,
species
no,
somewhere on
no.
the island.
No,
That's
no, no,
not
no.
nice.
A really brand new species.
But now you're letting other species go extinct, right?
Sorry.
Must be.
So you're letting other species go extinct.
Yes.
because you still
Yes.
exist.
In fact, that's the point. You have a turnover. If you can write down the master equation for this, and basically the interesting thing of this is that you can calculate explicitly all the correlation function. If you look at the one-point correlation function, that is what you were suggesting, then you're looking at that turnover. So if I look at this place, then you have a turnover. So this species sooner or later will go extinct.
In that particular.
In that, but you know that on average there will be another one that is coming in. So you don't go to an absorbent state, and you have basically your biodiversity is guaranteed by the fact that you have speciation, because always you have a little chance, a little probability that you have a new species that is in the system.
But in some sense, there is an average number of species that exist in stationarity.
Yes, there is. But like I said, I don't know now. I wouldn't.
Okay.
I don't know how to calculate it.
You have a saturation of the number of spatial sites, right?
Yes.
Can you
And
calculate-
always here. Sorry.
Can you calculate the species abundance distribution associated with this model?
That's another very good question, and unfortunately not.
Ah.
That's the point. But like I said, you can go from one, you can calculate some exponents related with the finance. And that's the reason why people don't use it anymore, because you can do something, but not much. Okay, if you can write down the master equation, the explicit master equation of this, and from this, I introduce this because basically what you can do analytically for this is to calculate the spatial turnover distribution that I mentioned before. So basically, you can calculate this. You can write down the equation for the probability that an individual placed at A and another place at B belongs to the same species. You can derive it from the master equation. Okay. So you see that this probability at n t plus 1 is given by this term, which tells you basically that you don't have to pick it before, pick either these two, or if you pick one of the two, basically the nearest neighbors has to belong to the same species that you go before. And of course you have this boundary condition here, because of course any individual is conspecific to itself at any time. Now, if you use the fact that we have what is important is the difference of the 2 points, so the distance between the two, you get this equation, which basically is the same as before, with the same boundary condition, and you can get the steady state, stationarity, you get this solution in dimension D, where L here is negative.
Sorry, I just on the previous slide, I was expecting there'd be a closure problem in trying to calculate this probability that it would somehow involve objects beyond, let's see, the two individuals may be more.
No, but the dynamics involve only its nearest neighbors, right?
Yeah, but if I try to calculate the spatial correlation function, which this essentially is, right?
Yes, but not exactly. This is a
It
probability.
seems harder than that, right? It seems
Yes.
harder than calculating a spatial correlation function. And we're
So-
talking, yeah.
So here, yeah, I mean, you could have something.
No, what I was thinking
I
was
mean, if nu is exactly zero,
right.
then you get this is the same that you get the same equation that you get in the usual Volta model.
No, I guess I was expecting that you would have like 3-point correlation, you know, like having calculated the probability of going on 3 sites at a time in order to.
No.
No? Okay.
No, no. This is one of the interesting points of this model, that
I
all,
see.
all the higher order correlation depend on the lower.
Okay, okay.
So it's kind of linear. You don't have a problem of a moment closure.
Okay, good.
Okay. Okay, this gives you the opportunity to fix. You see, when x is equal to 0, you have 1. So on the lattice, you fix. You have this correct boundary condition, but unfortunately, it's not good for dealing with the data. So what you do basically is to take the continuum limit of this. So nu goes to 0, sigma goes to 0, and goes to infinity in such a way that these two are finite. And you get this equation in the continuum when you escape time. And here, so basically this Dirac delta encapsulates the boundary condition and tells you that there is a non-trivial steady state. So if you go to stationarity with this, now we have to introduce this lower cutoff here, V1, because in the continuum space, you don't have information, or you don't have enough information to determine this. So basically, this C now is a kind of, or this V now is now a kind of new parameter that you have to use. Now you can do. It's easy to calculate this steady state, and you get this modified Bessel function here, which depends on this gamma minus 1, gamma to minus 1 is the correlation length. So basically, when R is very large, compared to gamma to minus 1, you have an exponential decay. Other than that, you have either an algebraic or a logarithmic behavior when R is smaller than gamma to minus 1. So, okay, this is what you can get for this model, very simple model. Unfortunately, this is not enough to explain the data, and the reason being the fact that you have basically we now know what's going on. We know that there is this Jensen-Connell effect. So basically, individuals, so seeds, tend to germinate far from from an adult conspecific. And the reason is that the individuals have this host, they have these pests that are host specific. So basically, this introduces in the model a kind of an effective speciation, so you have a stronger, a steeper decay. But this can be introduced straightforwardly in the model because you can define a piecewise constant, gamma. Of course, you have to introduce new parameters in here. But if you do this, you can, in 2D, you have this solution here, okay, C1, C2, you can determine by continuity. And if you apply, now you can apply this to see whether or not you get the data, and this is what you get. So basically, you see, you have these two scales here.
So how do you relate the survival probability of an offspring with the speciation coefficient? Is gamma or the speciation coefficient?
Yes, gamma is related to the, is not exactly,
It's not exactly
no.
the scaling.
No, because you see, the behavior of this model when nu goes to 0 and nu exactly 0 is completely different.
Yeah.
In fact, gamma is what you get in the limit, and not when it's 0. So you have a discontinuity in the behavior here. So what I'm saying here is that gamma is in the limit of nu that goes to 0, and basically what you are saying is that there is a probability that lowers conspecific to replace the adult trees. So you have this behavior here. So okay, this is what we know, more or less related to space and mean field. So what we would like to do now is, and this is some of more recent work, is to do this. So what we would like to do basically is not to model one pattern at a time, and this is basically related somehow to your question. So So not to model the species abundant distribution, the pair correlation function or whatever, but try to come up with a model from which you can derive everything. Okay, and this has an intrinsic interest, but it's also very important from the practical point of view. Because, for example, the species abundant distribution that I showed you before is very important for conservation purposes, for example, let me say just one. That is very difficult to measure in reality, because you have to sample everything, all the area. Here, the pair correlation function is much easier to measure, because you can just scatter some samples across the area, and then you get information. So if we had a model that can relate these two, from just this simple measure, we can derive the other. And so this has also important practical applications. So the idea, more recently, is to try to come up with this sort of theory. And to do this, always, okay, we are always referring to this one trophic level. We have now introduced these new things here. So individuals that belong to different species are more or less independent. This is what we already know. And on the other hand, individuals belong to the same species are spatially clustered. So they are aggregating in space. So
But doesn't that contradict what you said, that the new offspring goes away from where the adults are?
No. What I'm saying here is that it's not a contradiction. It's that it just lowers the effect, but aggregation is always there.
Okay.
So it's lower with respect to the model that I've shown you before. But in general, you can model it in many different ways. So the idea is, again, to have a very simple spatially explicit model from which we can derive these patterns. Okay, so what I try to do is to introduce a metapopulation model, so basically a regular graph in which at each and every site we don't have just one individual, but many individuals. And the dynamics is very much similar to what we see before, but also different. So an individual in each and every site can die at rate d. The birth, instead, of an individual occurs at rate b, and in addition, a newborn can hop onto one of the nearest neighbors with probability 1 minus gamma, and remain in the same site with probability gamma. This is not enough to have a steady state, but if you add immigration, you have the steady state that you need. Okay? So basically, this is just a constant rain of properties that affects the entire landscape. Okay? This is simple enough to do some calculations and to do some predictions. So you can write the total birth and death rates here. So you have this birth rate and the individual stays there. You have aggregation here and immigration and the death rate. So basically what we are seeing here, that if we succeed in predicting this pattern, basically these are due to demographic stochasticity, diffusion, and immigration. This is basically the basic ingredients. We don't have no linearity, so in general B is smaller than D. We have spatial aggregation, and to a certain extent I would say that this is a minimal model, because if you get rid of one or more of the parameters, you get something that is completely useless. Okay? You write down the master equation even in this, and you can use the generating functional, or if you want to call it the partition function, whatever, if you like statistical mechanics. And basically the master equation that I've shown you before turns in this equation for the partition function. And in the continuum limit, we have this equation in dimension d, where h in this case is the conjugate field to the field of the population. So here we have diffusion, birth and death, and immigration.
So can I ask a question about the model? I just want to make sure I understand. So go back a few slides, just where you're describing it, right there. So death is clear. Someone at a site dies.
Yes.
Birth. So someone here has an offspring.
Yes.
Goes to a nearest neighbor.
No, with probability 1 minus gamma, it stays where it was born.
So, but is this only if? Sorry, I'm confused.
No, fine.
Is it only if the site is empty that there
No,
can be a
no.
birth? Okay, so if there's a type of tree here.
No, no, okay, if the, of course, if you don't have individuals, then you cannot have,
Okay.
yeah, but if you have individuals, then you have this bird.
Is
So
your
you have
density
multiple occupancy.
increasing?
Sorry?
Is your density allowed to increase?
What do you mean? No, the number of individuals in a site can increase and decrease, they fluctuate, yes.
That's what I was thinking. Okay.
So it's exactly like a reaction diffusion mass equation.
Other question? No.
But really, because the diffusion is linked to inverse.
Yeah,
But
but the
it's
difference
zero.
is that here you don't have no linear terms.
Right.
You don't
So in
need
some
to-
ways it's simpler, in some ways
Yes.
it's more complicated.
Yeah, so, okay, so I get it now. So if there's a, I'm gonna say it again, though, just to make sure I get it. So if there's a birth, it can go up where I am
Yes.
with probability gamma or
Yeah,
one minus
whatever, can hop.
It is born, but then is
Yes.
placed right here.
Yes. Yeah, and of course you can use not only this diffusion, you can have a kernel, whatever. It doesn't, of course, yeah, whatever. Okay. Okay, this is the equation in the continuum limit. To get a flavor or of the prediction that this model does, I use this mean field prediction here. I mean, if you get rid of the spatial degree of freedom, you get this Fokker-Planck equation in the diffusive approximation, where you have this n here that is 0 at 0. So it's kind of interesting equation here. And this is basically due to the central limit theorem, right? If you think of the Langevin equation, this is a square root of n. So basically this is central limit theorem, birth and death and fluctuations, okay? So this is the main ingredients of this. And when I got this equation, I was very happy because I knew what one can do with this equation, because you can calculate basically everything. You can calculate not only the steady state, you can calculate the full time dependence at with absorbing conditions, reflecting boundary condition, whatever you want. Basically, you can do whatever you want here. But this gamma distribution here is what you are interested in, because this gives you this species, the relative species abundant distribution that I mentioned before, the probability that a species has n individuals. Okay.
So can you remind me, is this diffusion, the gamma, does that affect sigma?
Sorry, say that again.
What is the role of the diffusion gamma?
No. In here you don't have diffusion, right?
So gamma's just gone, it makes no
Yes.
difference.
Here there is no space here. Sorry. Okay, if you look at the mean number of individuals per site, species, of course this is a kind of trivial behavior, and this is the reason why, I mean, it's not exactly a reaction diffusion equation, because everything is, it's very simple, the mean behavior of this. And you see here, a stationarity, you get just everything that is constant. So quite boring. So what this model explains, if any, is just this. What it predicts because of fluctuations. So basically, patterns that emerge from this model emerge because of fluctuations. So let's go ahead. You can calculate the two-point correlation function at stationarity, and you get this this equation for the two-point correlation function with this Dirac delta here. You can solve this, okay, and you get this distributed with k naught, again a modified Bessel function with these two spatial scales here. Okay. The interesting thing of this model again is the fact that you can calculate all the pair correlation function at all orders simply because it's linear. So you can calculate the endpoint correlation function. But unfortunately, the problem here is that when you look at the species area relationship, you don't need only to know all the endpoint correlation function at all orders, but you have to be able also to sum them up. So basically, you need the full solution, the complete solution of your model, simply because this is a non-local quantity, a non-local measure. This depends on all pair correlation function at all orders. And this pi here is the is a conditional probability. The conditional probability that a species has n individuals, given that we are looking at an area A included, that is within the area A naught. Okay? So basically, if we want to calculate the species area relationship, we have to be able to calculate this pi of n given A. And this is basically the non-trivial task, the non-trivial pattern that one has to calculate. And this is also called the spatial relative species abundance, or the conditional relative species abundance. You can see what are the predictions for this, the distribution of the model. So you get this conditional partition function, and the equation is, you can get the equation for zeta of A simply by substituting this indicator function. So if you substitute this into the original equation of the for the partition function, you get this equation for the conditional partition function. So basically, this is the partition function for the conditional probability that you see a species with n individuals within the area n. So, and in the continuum limit, you get this integral equation, coupled integral equation in here. So my idea at first was basically to look at the steady state of this. So basically, I thought, okay, it's enough if we focus on this integrand in here, and we derive, we solve the model here, and then we see what's going on. So we can, okay, if you do that, you are completely wrong. It's completely wrong. The reason why, and it took me a while to understand this, the reason is that this the model that I show you here is a no-equilibrium model. So you can have stationarity, but it's no-equilibrium. That means that detailed balance is not satisfied. Now, if you insist on saying, okay, we impose detailed balance to this model, let's see what happens. If you impose detailed balance, you get exactly the integral. So basically, you get this equation here. And if you try to, so basically, if you look just at the integrand, you are imposing detailed balance even though it's not correct, it's not satisfied for them. If you go ahead, you insist on this, you get a 3-point correlation function that is not invariant under swapping on the labels, which is a nasty thing, right? And even the probability that you derive from this rock here gets negative values. So basically, you can't do anything. So so here we were stuck. But what I did instead is try to introduce an approximation. And the approximation is as follows. So basically what we do is this. Of course, an approximation to calculate this conditional probability, of course. So we start from the steady state of the mean field. So we get rid of the degree of freedom. And in this case, we know that it's just the RSA, is just the gamma distribution. Then we calculate, so we look again at the spatial model, so the stochastic PDE. We look again at and we solve just the 2-point correlation function, so just the correlation function. And in this case, it's just this. So the steady state of the mean field, and now just the 2-point correlation function of the spatial. From this, we derive the spatial variance of this model, and then what we did is this. So alpha and beta are these two parameters. So we substituted to alpha and beta two functions, function of the radius of the area which you are looking at, and then we imposed a moment matching basically. So we imposed that the first moment and the second moment are right those of the original stochastic model, okay? But, of course, we use the approximation that the RSA is the one given by the mean field. The interesting thing is, okay, you get these complicated coupled integral equations, but in this case can be solved. This is the interesting bit. And if you do that, basically what I got is this alpha and beta. Yeah, they are nasty, but okay, who cares? I mean, but the important thing is that these are analytical calculations that so we can exploit all this in the future, and I will show you the data. So basically, now we succeeded in getting this approximate conditional probability distribution with these two functions here, which depends on three parameters. You see, B over mu, rho, and lambda. And now, of course, with this conditional probability, we can link all these patterns.
So particular alpha times beta is always some nice thing, right? All the nastiness goes away when you multiply alpha and beta, is that correct?
Yes. Yes. In here, you link all the patterns that I've showed you before, even though with this approximate, in this approximate way, but you can link them all. I tried to do simulations of this, and I don't have in my computer here, but if you want, I can show you. There is a regime in which this is a very good approximation of the conditional probability. So there is a regime in which this exactly, so the idea now is to, because here you have to use a singular perturbation for a stochastic PDE. That is what I want to do now in order for me to control this approximation, but can be done. Now let me show you what's going on with the data. So again, I use the data from the Barra Colorado Island, and I apply the model to the data to see whether or not it fits the data. Okay, first of all, the mean number of individuals as a function of the radius of the sample area. Okay, this is trivial to a certain extent because it scales with area. The number of individuals scales, so nothing to say about this. But okay, I'm not fitting any parameter because n is the total number of individuals in your plot. S is the total number of individuals in the plot, and A is the total area. So there is no best fitting here. Where the best fitting comes in is here, where we have the pair correlation function here. So here I best fitted just the pair correlation function, and so I got this rho and lambda. I just best fit and got rho and lambda. But now, I fixed all the parameters of the model because I have three free parameters. I fixed everything, so I can make some real prediction about what's going on the other patterns. That means the species area relationship, the relative species amount, and so on. So I can do all the prediction I want. And here, so with these parameters that I got here. So this is what I got here for the spatial model. So these black dots are the empirical data, and the red line is the model, the prediction from the formulas that I've shown you before. Whereas the random placement model is this systematic overestimation here. So basically, I'm almost always within the 95% confidence interval. With the same parameters, now I predict this is the species area ratio. I predicted the relative species abundance, and this is it. This is the curve that I obtained for the species, for the prediction for the species abundance distribution at the entire plot. So at the large scale. And you see, I'm more or less always within the 95 confidence intervals. But I went on and I tried to see what's going on at finer scale. So it kind of downscale. And you see, the model is always able to keep track of this downscaling of the species abundance distribution. So if you go down, you really get, you have a good understanding of what's going on, at least at this. Here we are predicting the behavior of many different patterns with same three parameters, okay, that we got as stationarity. Now it's 1 hour. Can I go ahead? Or
I mean, you can have 5 minutes, I guess, 10 minutes. I mean,
yeah, five.
Otherwise, if you don't want to.
No, that's fine. Okay, I can 5, 10, whatever. Okay, this is, and this also, if you play with the parameters, not exactly, this is not a real prediction, but this is just playing with the parameters of the model, you can also get this triphasic behavior of the species abundance, the species area relation that I showed you at the beginning. You remember?
I thought that the opposite went from steep, not steep, to steep.
Sorry?
Didn't it?
Sorry?
The earlier plots?
The top right is steep, not steep, steep. The top right is steep, not steep, steep.
Jay is
Well,
referring to
it's
the original.
four-phasing.
Yes, for phase.
Oh, I see, the last punch out.
Yeah, but this here, you mean?
That one most closely matches the earlier plots. This is a question.
Yeah, no, but like I said, this is not. I'm not. These are different parameters. I just wanted to see whether the model with the some combination of the parameters can have this kind of behavior. I'm not saying that the model is able to predict the true triphasic behavior. I'm only saying that there is some combination of the parameters for which you can see this. I don't know whether this can fit what I showed before. Now, let me go quickly on this. So the approximation method that I use for this stochastic PDE can be used regardless of the model itself, because one could say, okay, if you give me the species abundant distribution and the pair correlation function, then from this you can, without doing any other approximation, you can calculate the spatial variance, and then with the same trick, with this moment matching, you can get alpha and beta. But in this case, you are not assuming anything about the species behavior. In this case, they can interact, they can do whatever you want. You are just phenomenologically trying to predict what's going on. And this is, okay, we don't assume anything about, we need, okay, abundances and whatever, but we are not assuming anything about the neutral behavior or independence behavior that I showed before. Why is this important? Because this addresses one of the hot open questions in the field, which is the so-called upscaling problem, which is easy to state. So imagine that we have the Amazon forest here, or we have a large area, and we want to give an estimate of the total number of species. Okay, number of species is subadditive with the area, so right? So the idea is that you can't survey everything. If I give you random samples, scattered random samples over this landscape, what should I do in order for me to extract the information and predict the total number of species that live therein? This is basically the question. Of course, look at the Amazon forest, and these are even, it has a recent interest here. So look at the Amazon. We can't survey the entire Amazon, but what we can do is to have some scattered samples of reasonable size across all over the Amazon. It's still a tiny fraction of the entire area, but it's still something. And of course, we know from this, we can calculate the average number of species per site, and we can calculate the pair correlation function, the 3-point correlation function, so on and so forth. So do we have a way, a technique, a method, whatever you want, from which all this information can be collected in such a way that we can predict the total number of species that we have here? Basically, so far, there are many models, but none of them, or nearly none of them, uses spatial information. So, you agree that you are losing lots of information here. So basically, they lump everything together and they try to make a prediction. Of course, this is not true because and in fact, you can see in many examples that this is not true, because you're throwing away all the information that is encapsulated in the correlation function. So this would be true if species are distributed at random, but they are not. Okay? So I tried to do this exercise here, but not for the Amazon forest. I don't have the data for that. I tried to do it for the Barra Colorado Island and for the Paso forest. And so these green dots represent the information that I used. This black line is basically the prediction. But of course, this is a kind of an exercise because I'm not using a huge scale, a huge upscale, right? It's just 1-2 order of magnitudes, not more than that. In the Amazon forest, we have to say 6 or 7 order of magnitudes. So it's really a challenging task. But you see that from this we can predict the species abundant distribution at the entire scale. So we are not only saying how many species are present in the entire plot, but we are even saying how abundant we will find the species that are yet to be observed. How much abundant. So, okay, I try to do it with France, with breeding birds. Just an exercise to see whether or not it works. And in here, I did the same exercise. And the prediction was fairly good. Here, we have more or less 4 order of magnitude. So we are kind of, and the expected number of species in France was 310. And this is our prediction. So now we are still working on this, and probably we need much more work. But at least these are kind of open questions, some scattered open questions that are related to this macroecological scale. So, okay, so we are at the end. So there are many things. For example, I would like to, if possible, I would be very interested in finding an analytical solution for the spatially explicit model, for the stochastic model. But basically, this is a good approximation of that is this. If you think of, for example, animals, like this. This is it's not exactly what I've showed you in the original model, but this is the diffusive approximation, and in some regimes it's well approximated by this stochastic PD. So if we could, to a certain, and on the other hand, this can be thought of as a linearization around the point of an underlying theory. So this would be really good. It would be really good to have the partition function of this model. So far, I've got some insights of this. I can do some, like I said, some singular perturbation theory on this term, and it works pretty well. Yeah.
What about doing singular perturbation on the full model like WKB?
Yeah, yes, that is another option. That is another, but
It might be harder than this, obviously.
But
If you have anything that relies on tail events, or
but I should identify a parameter that.
it's whatever takes you to your deterministic limit that you were talking about.
Yes,
sigma.
yes, but the problem is that in here, fluctuations are important. So I'm not looking for a deterministic limit.
No, I know that you're perturbing off the deterministic limit, so the WKB would give you the stationary distribution. It's sort of like exponentially accurate as long as your parameters.
Yes, but close to a deterministic limit. Yes, but what I'm saying is that the behavior that I show you here so far is not, I'm not close to a deterministic limit. The perturbation effect is this here. I'm close to a kind of mean field limit, but where fluctuations are important, are very much important. So
I think we're in violent agreement. I think that the
it's
noise terms are going to be really important.
Yeah.
But the stationary distribution from which you get that conditional distribution that you wanted, you could get that. I think in a way you're already sort of doing this singular perturbation off the mean field.
Yes, to a certain
That
extent.
would just be a more formal way of
But anyhow, these are open question and future work for not only for me, for many other people. And if you're interested in these kind of things, these open questions, this work, a little advert, here. It just came out, this paper here, which basically summarizes what has been done so far. Not everything that I show you here is in this paper and vice versa, okay? Okay, sorry for the delay, and thank you for your attention. Yes?
Yes.
Just some stray thoughts, maybe following somewhat in the vein of what Jay was suggesting. So I sort of have three stray thoughts about kind of just the methodology. So one is, I believe if you take space out of this, then in finance, this is called the Cox-Ingersoll-Rand model, is that right?
Yes.
Okay. And so one thing you might do if you're trying to get an exact solution is maybe someone's looked at a coupling of two assets that are each modeled by Cox-Ingersoll-Rand process, and is there a possibility to extend the stationary distribution to that, right? That might be one place to start. But then it's sort of more in the asymptotic lines, so like what Jay was
Yes,
talking about. I was trying to think what, okay, so one thing that what you're doing reminded me a little bit about was Gaussian copula. It's not that, but it sort of sounds like it in the sense that.
Yes, in a certain.
Okay, you know what I'm saying. And then and the other thing is it also feels a little bit to me like when you do like hydrodynamic limits of microscopic systems, right? Okay, so.
Yeah, that's the point, yeah. But in here, to a certain extent, we need non-local information. We need, at least in some regime, not always, but in some regime, we need much more information.
And then what's hard is that you sort of have like your variable that's kind of your slope parameter. It isn't just like, you know, time or space. It's like this radius that's accumulating information from before, so it's a little bit trickier.
Yeah. And that's the reason why it's not so easy to get it. But on the other hand, if we succeed on this, we could think like a kind of linearization, a stochastic linearization around an underlying nonlinear theory, and that might be really useful, not only in ecology or in this, but even outside.
But how do you formulate
what
like
I'm
this?
trying to do now is, and that probably is useful, is to use path integrals in you know, the statistical mechanics, the field theoretic approach.
There's this Carty stuff.
No, the you know, with the fog space where you have this ladder operator.
Exactly. Yeah, that's right.
And okay, we are
That
referring to
was the
different
part
things.
2 of my talk I didn't get to.
Okay, yeah. And this, because in this case, you can decouple space and you see what I mean. You can decouple basically if you write this basically as a matrix, you can decouple the row and columns to a certain extent, and that might be a good way to get the final answer.
Does that field theory give you the full probability distribution, or just?
In the linear case, yes.
Okay.
In the linear case, but you see, if I can get the answer there, I have already the machinery for the perturbation approach, and so I can add here nonlinear terms at will, and I get the right answer. And by the way, for example, if I multiply this by n, I got the direct calculation theory for the region field theory, right? So it's kind of, this is kind of in the middle of different regimes like you noticed. And might be interesting to see whether we can link from this point of view this approach.
I suggest we stop here and we can continue over tea.
Yeah,
Very
okay.
much, Sandro.